Assalamu alaikum. Myself Laraib Elahi and playing a role of presenter in Sin Biotechnologists Association. Welcome to my presentation. As you all can see on the screen, my topic of presentation is designer babies, a healthcare wonder or an ethical error. So let's start with the presentation. Designer babies are basically genetic engineered in vitro. In vitro is a Latin word that means within the glass. Anything which can produce outside the living organism, it may be produced in a petri dish or in a glass tube. They carry specific traits that we should want to be transferred in a baby. These specific traits carry lower disease risk as well as the genders of the babies can also be selected through these techniques. The designer babies is an embryo. Embryo is basically a group of cells which has been fertilized or implanted in the uterus that will become a baby in the future. These designer babies have done genetic makeup on it for making them with the specific traits. We can select the eye color, we can select the hair color, we can also select the height of the children by making them designed by doing genetic makeups on it. Is world first designer baby reality? Yeah, it is a reality because this treatment was being successful in the past, and also scientists are working on it. The proof of this reality is Adam Nash. Adam Nash is the world first designer baby, which was born in US in 2000. He was free from Fanconi anemia. His family was has a inherited genes that carries for the baby and hence for sister. His sister was suffering from Fanconi anemia. It is a genetic disorder in which bone marrow is defected. This results in decreased production of all kinds of blood cells. So Adam was produced for saving his sister's life from these genetic conditions. So the treatment became successful when the stem cell was donated by Adam to the Molly for making his life safe. So Adam Nash was called savior of his sister. Lulu and Nana. Lulu and Nana was for gene edited human babies. These gene edited babies were produced by He Jianko in 2018. He was a scientist of Southern University of Science and Technology in China. He produced these twins girls, which named Lulu and Nana, by the help of a technique, CRISPR-Cas9 genome editing technique. It is basically a protein which can alter the defected gene. The defected gene is removed from the human DNA and replaced by the altered gene. So these babies were produced to overcome the immune to HIV virus which were inherited from their father. These are the techniques which are used in producing or altering designer babies. In vitro fertilization, pre-implantation, genetic diagnosis, and germ gene editing. Further, I will tell you more about these techniques. Fertilization. It is basically a technique which is done outside the living organism for producing a designer baby. The first step is ovarian hyperstimulation. It is basically a induction or inducing medications or injections to a woman for producing eggs. After the eggs are produced in the woman, the egg retrieval is done. Egg retrieval is the process from which eggs are being taken from the uterus of woman. After egg retrieval, sperm and eggs are prepared in the lab in a petri dish for further examination. After some days, 2 to 3 days, these eggs became fertilized. These fertilized eggs are examined. If they are healthy, then they are transferred to the uterus of a woman. After the transfer of a embryo, after the passage of some weeks, 3 to 4 weeks, the pregnancy test is done if the embryo is fertilized or not. After IVF, pre-implantation genetic diagnosis is done. It is also a technique which is done soon after IVF. Embryo is cultured from the sample of IVF. These cultured embryo is go further for biopsy. Biopsy is a process in which a group of cells or 1 to 2 to 3 cells are taken from the embryo for testing. If these embryos are produced or fertilized, these are moved forward for tubing. Tubing is a process in which these embryos or cells are being put in a tube for several hours. After the passage of hours, these culture is furthermore for analysis. Genetic analysis is basically a technique which is done by scientists or lab examiners in which they observe the genetic materials if the genetic makeup of the cells is okay or not. If the cells are carrying a diseases or any other mutation. So these defected cells from the embryo is being removed in the process of evaluation or result. So the healthy cells are being transferred again to the woman uterus. Third one is germline gene editing. It is basically a simple process in which gene editing is done. The defective gene is being cut from the part and it is repaired by altering or inserting a healthier gene into it. As you all can see, this part is cut of DNA. After the part of DNA is cut, it is inserted with a new gene that is called gene editing or gene replacement. It is the process which is done in labs. The cells from the patient suffering from any diseases or viruses, the cells are taken from the patient. After the cells, these cells are moved for labs analysis. The cells can get altered or produce these viruses to overcome the issue. A gene is inserted. In the third step, a gene is inserted into the virus. If a person is carrying any virus, so a gene against the virus is inserted. As you can see, it is representing the purple DNA. These DNA strand is replaced with these blue coded DNA for alteration. These altered virus is mixed with patient cells. Virus cells mixed with patient cells. After they are mixed, the cell became genetically altered. As you can see, the mixing of two genes, the virus genes and the human cell genes, they when they together mix up, they form a genetic which is altered, a gene which has done genetic makeup on it, which is genetically altered. After this, the altered cells or gene is transferred or injected into the patient. It can be injected in many ways. So these injected genes, altered genes, are then further transferred into the human body and it will work as a time period will pass, they will produce a desired protein which the person needs. So it is basically a gene editing or the germline editing. Tools which are used for the modification of genes. There are many tools which are used for the modification of genes. The main tools are CRISPR. It stands for clustered regularly interspaced short palindromic repeats. This means it is the tool which is used for modifying the genes, altering the genes. These technology is working nowadays. Scientists are using these technologies for altering the defected genes. A human found with defective genes can be cured by having worked on DNA with the help of these tools to overcome these diseases. So, this is basically a CRISP which has three components: guide RNA, which guides the DNA from which part which is defected and from the part where the segment should be cut. Matching genomic sequences. It is basically for joining the sequences with the correct sequence. As you all know, if the sequence is not correct, it is not matching, the sequence is not matching with one another. So, as you know, the mutation will occur in it because the sequence of the DNA should be in proper manner. So, if this part is showing the empty space because there is found a disease or any defect or the mutation in the DNA. So this part of the DNA is cut and inserted with a new gene which is called a program gene. So this being part of the segment of DNA is inserted into the DNA segments to overcome defects, to overcome the disorders, and the gene which is now made is altered gene. On the other side, CRISPR associated protein. It is a technique which is used for germline editing and it stands for associated protein 9. These enzymes are working like a scissor. They are cutting the parts of the DNA, the segments of the DNA, which the guide RNA guides to cut. So it is working like a scissor in the body or in the DNA. It contains three main components, which is the associated protein 9, which is working like a scissor, and on the other side, the guide RNA, which is targeting the mechanism from where the point or where the part should of DNA should be deleted or inserted with a new segment of DNA. So they together combine with one another and produce a new prescribed surgeon, which is called CRISPR molecular surgeon or CRISPR associated protein. The working of CRISPR protein is when the protein formed in a complex structure. These DNA structures are complexes and joined with guide RNA, ribonucleic acid. These are ribonucleic acid which guides the CRISPR or associated proteins to bind with the genomic structures of the DNA, where the sequence they found should be adjusted at a part of a yellow segment. As you can see on the screen, this yellow part is found for the attachment with another genes which should be inserted. This part is found as a defect or mutated in the genes. So these part of DNA is cut and inserted with the programmed DNA. These programmed DNA is inserted into the DNA segments. So these DNAs are able to produce proteins in the future. Designer baby is a healthcare wonder because parents can change the gender of a baby. They can produce a baby boy or a baby girl as they wish. They can choose a favorable characteristic which they want in a child. They can increase the IQ level of a child. They can produce a baby with brown hair color or the eye color, or the vision of the baby can also be selected by doing producing these designer babies. They can help in preventing the genetic diseases which are not produced in the future baby. They can overcome these diseases before the birth of the babies by making alternation in the genes and DNA. Mitochondria, as you all know, is the powerhouse of the cell. If the cell or the powerhouse will not work, then the DNA will not produce the proteins. So these disorders are prevented from getting defects. Also, it will increase the life expectancy because the DNA of the baby is already altered, already modified, so they will not have any chance for producing any disease in the future. It will provide the positive influence on the children and on the society, as they we can also control the lifestyles of our children before the birth. So it is all un- under the genetic engineering. As peoples are getting more toward these genetic engineering, peoples are producing designer babies, so these technology is successful nowadays and peoples are using these technologies. On the other side, is the designer baby an ethical issue? I think it is an ethical issue because there is a large gap between the rich people and the poor people, because this technology is very expensive. Poor people cannot afford it. They cannot modify their baby before the birth. So, rich people will modify the baby. They will have the babies with high IQ level, with the absence of genetic diseases, with no defects will found in the genes of the rich peoples who will produce designer baby. So automatically this will make a large distance between the poor people and rich people, which is a big issue regarding designer babies. It will also harms the mother's body, because when we take embryo from the mother, it is very difficult. So, when techniques and further investigations are done on the embryos under lab examination with the help of IVF, with the help of PGD, so automatically, when these embryos after alternation is inserted into the uterus of the mother, the mother gets hurts. He he she feels pain, so that's why it is difficult for the mother. So I think it is harming the mother's body. On the other side, the gene pools also may be affected by doing these techniques, using these techniques and producing a designer babies. Some genes which are essential for the growth of human beings, which are needed in our life, it can be removed by using these technologies. The more we work on it, more we get experiments on it, so these genes will automatically will be removed from the DNA, and the DNA will not have the genes which are essential for the growth of the human being. It's my opinion, or you can say it is unethical. I think it is unethical because it is harming the society in many ways, making large distance between poor and rich people, affecting the mothers, and somehow it is affecting also. So it also have advantages. On the other side, it has many disadvantages. So it's up to you if you can afford it or you can consume these technologies, so it's up to you. Ethical and moral debates are doing day by day. World Health Organization is also taking steps against these. So human germline engineering should be practiced or not, it is a major concern about the debate. It is ethical or unethical. This topic is very hot nowadays because people are opposed to use these techniques. They believe they will create the human with all the perfections, they will create a human beings with no diseases, with a high life expectancy, with a favorable traits, and many more. So this will lead to the social gap. As I said earlier, this will lead to the social gap. So they had benefit also and disadvantages also. So the altercations between the scientific community and the global community regarding this is debating more day by day. Legal and regulatory frameworks in different countries regarding gene editing, producing designer babies are done. According to the survey of 2016, the scientists examines the laws that can be documented by the policies or by the governments. So the gene editing should be done or not. It can be applied or not applied to the human being. So the scientists are searching on it. Many scientists are, and many countries are having legal framework. Some countries are restrictive towards using these techniques. Restrictive countries includes Canada, Brazil, Germany, Australia. These countries are restrictive against gene editing and producing a designer babies. On the other side, some are permissive, like China. As you all know, China has already produced the first gene edited twins babies, which was Lulu and Nana. So UK is also working on it. Many countries are working on it, and some has against toward these. So these techniques should be used for the betterment, for making and modifying gene alteration for the humanity, not for disturbing the society, making designer babies and making the wide gaps between the poor and rich. The conclusion for this is genetic engineering is, as you all know, is a branch of science which is concerning about the all the genetic engineering, the makeup of the genetic material, the alternation in the genes, the selections of the genes can also be done. So a baby with a complex DNA can also be produced by it, but somehow it may face some issues by producing a designer baby. So this technology is somehow a risk, or somehow it is an ethical issue or unethical. So it depends upon the using of these techniques. If we are using these techniques in a right way, so that's okay. If we are harming the society, we are harming the mother bodies, we are making a child perfect, making the child high IQ level, so this will lay down the other person, so they are defecting like this. Safeguarding human dignity will be crucial in the future, as I already said before. The debates are continuous day by day. As these technologies are increasing, the debates are also increasing day by day. Peoples are concerning more about it. Some peoples or some countries are having legal action towards it. Some are restrictive and some are consuming these technologies. So it is best for the interest of humanity mind. So it is just for the knowledge, not for consuming these treatments for the bad or wrong things. Further, move on the references. These are the references from which I collect the data about my presentation. As you can see, these parents are going to buy a baby, designer baby with their own characteristics. They want a baby with funny cartoons, or they want a baby who will become the tax lawyer in the further, an actor or fashion designer. So parents are also selecting these types of babies for futures. So it's up to you. It's a depends upon the country legal actions. They can take legal action against it, and on the other hand, some countries are producing these babies, so it is somehow affecting, have many advantages and disadvantages. I hope you like my presentation. Thank you for listening. And at the last, I would like to thank Sin Biotechnologies Association for providing such a platform where I can share my knowledge with you all. Thank you so much.